Although DNA was first discovered in the late 19th century, it was not until the middle of the 20th century that the role of DNA as the genetic material was established. Years of research were necessary to come to this conclusion. Frederick Griffith, a British scientist, hypothesized that information could be transferred between different strains of bacteria. He established that there was a transforming principle in bacterial genetics. According to the Merriam-Webster dictionary, the transforming principle deals with DNA that is transferred from one individual to another in genetic transformation. And to clarify this further, according to MedicineNet.com, genetic transformation is a process by which the genetic material carried by an individual cell is altered by the incorporation of foreign DNA into its genome. Griffith didn't know the chemical and biological processes behind the transforming principle, so he did inspirational research based on the theories of scientists like Mendel. In 1928, he conducted an experiment on mice using two different strains of Streptococcus pneumoniae, bacterium that causes pneumonia. The outer coat of the bacterium is either smooth and virulent, the 3S strain, or rough and non-virulent, the 2R strain. For the first stage of the transforming principle experiment, Griffith showed that mice injected with the 3S strain died, but when injected with the 2R, they lived and showed few symptoms. The next stage showed that if the mice were injected with type 3S that had been killed by heat, the mice all lived, indicating that the bacteria had been rendered ineffective. The interesting results came with the third part of the experiment, where mice were injected with a mixture of heat-killed 3S and live 2R. Intriguingly, the mice all died, indicating that some sort of information had been passed on from the dead 3S type to the live 2R type. Blood sampling showed that the blood of the dead mice contained both live type 3S and live type 2R bacteria. Somehow, the type 3S had been transformed into the type 3R strain, a process he christened the transforming principle. In 1943, Oswald Avery, Colin MacLeod, and Macklin McCarty performed experiments on mice, building off of Griffith's works and findings. Their experimental demonstration reported that DNA is the substance that causes bacterial transformation, in an era where it had been widely believed that it was proteins that served the function of carrying genetic information. These three men put strain 3S bacteria into a test tube and added protein-destroying enzymes, which did not work, and so the 2S strain was still able to transform the 2R strain. However, when DNA-destroying enzymes were added, the 2R bacteria couldn't be transformed, and they concluded that DNA had been the transforming agent. Alfred Hershey and Martha Chase worked with bacteriophages, small bacteria-infecting viruses with an outer protein coat that surrounds DNA. They knew the viruses were transferring hereditary information to the infected bacteria, but weren't sure if the information was protein or DNA. In 1952, they infected bacteria with viruses that had either radioactively labeled proteins or radioactively labeled DNA. Infected bacteria became radioactive only from the DNA, proving that DNA is the molecule of heredity. Maurice Wilkins, Rosalind Franklin's lab assistant, began using optical spectroscopy to study DNA in the late 1940s. In 1950, he obtained the first clear crystalline X-ray diffraction patterns from DNA fibers. With X-ray diffraction, as X-rays pass through the molecule, they bounce off before striking the photographic film. The patterns produced on the film by those bouncing X-rays can be used to reconstruct the 3D structure of the molecule. Franklin's images of the DNA molecule ultimately revealed its exact structure. James Watson and Francis Crick wanted to determine the 3D structure of the DNA molecule. To do this, they looked at Franklin and Wilkins' X-ray diffraction images without their knowledge. They figured out that DNA is made up of two mirror image strands composed of stacked DNA nucleotides, which are made up of a sugar, phosphate, and nitrogenous base, linked together into a twisting double helix. This double helix can be thought of as a ladder with deoxyribose sugar and phosphate forming the sides. Adenine, thymine, cytosine, and guanine are joined by weak hydrogen bonds and form the rungs. A always binds with T, and G always binds with C. The order of these molecules represents the genetic sequence. For this discovery, Watson, Crick, and Wilkins were awarded the 1962 Nobel Prize in Chemistry. Matthew Meselson and Franklin Stahl conducted an experiment in 1958 which supported the hypothesis that DNA replication was semi-conservative. In semi-conservative replication, when the double-stranded DNA helix is replicated, each of the two new double-stranded DNA helixes consists of one strand from the original helix and one newly synthesized. They placed the bacteria, E. coli DNA, in an environment which contained a selected nitrogen isotope. The first used was nitrogen-14. The bacteria then integrated this isotope into their DNA. Later, they used an environment that contained nitrogen-15. They then looked at which of the isotopes the bacterial DNA contained. They found that it contained both isotopes of nitrogen, which implies that conservative replication is not the correct conclusion. By having a closer look, they could then also rule out dispersed replication. Some helix strands were all nitrogen-14, while other helix strands from the time in the nitrogen-15 environment had nitrogen-15. Thanks to the years of research and the dedication of these scientists, the discovery of DNA's role as the genetic material was made possible.